So it was a really good experience at Roskamp. We got to work with not only NASA scientists and not only the staff at Roskamp, who were really fantastic, but we also got to work with several lead scientists attached to the RR20 mission. So it was really a fulfilling experience for myself, but also for my lab. My lab's actually accustomed to these kind of dissection days where you not only take muscle, you're taking other tissues like brain, adipose, bone. Not that we are doing the work directly on these tissues, but we are getting them for another group on this floor. So when we went to Roskamp and when we worked with NASA, it was the same idea. It was, you want to maximize what you get out of a mouse that just flew up into space. So there's a group there that's specifically focused on muscle for us, for example. There's another group focused on bone, another group focused on adipose tissue and brain and what have you. And so when they asked us if we can help collect this and this and this, kudos to the two students from my lab. They already knew that, hey, look, I can do this. So that was a really cool moment for us to go in there and show that we can collect our data that we want to collect, but also help out with the other projects that are ongoing. And I think really it bolstered their networking, their network. And I think that's really important for enriching their training, the training that they receive, not only from my lab, but from others across the globe. The initial goal of this project was to look at the effects of spaceflight on female fertility. So we know now that as the years have gone on, the proportion of female astronauts have gone up, which is very important and really good. However, we still need to understand the physiological and biological impacts of spaceflight on females. And so this particular project, which was led by Dr. Ling Christensen, focused on fertility. So what happens to these female mice when they go up to space? Do they become infertile or not? Turns out they're okay, because we were able to look at the F1 pups. But that was the original goal of the project. And where we came in was, our goal really was to look at muscle contractile function, because no one has actually done that. Again, what has been typically done in the past with NASA is they will take the tissues, they'll take the muscle, and they'll store them in this bioarchive. So basically in a -80 freezer, and wait for other researchers who are interested in assessing these muscles from a biochemical standpoint. But there's no functional measures tied to it. So that's where we want to fill that knowledge gap. We want to go in there, provide some muscle contractile data, give the muscles back to NASA and say, you can still do your biochemical work, but here's some functional data now. My initial goal, aside from the science, was to go in there and show what we can do and show that we can work as a team. And I'd like to think that it was successful. We were invited another 2 times after the first time in December and January to travel back down to Florida to collect some more contractile data, but also to help out and collect some other tissues for the other NASA scientists. So I'd like to think that in terms of progress, it was really successful, and we're working on some other projects as well. So what we found is basically bang on with the literature and what you'd expect. So with spaceflight, what happens is you unload the soleus muscle. The soleus muscle is this postural muscle, so it helps you stand right here in Earth, right on Earth. So you are withstanding the never-ending downward pull of gravity. But when you go up in space, that soleus muscle, this postural muscle, becomes unloaded. And what happens is it experiences what we call a slow to fast fiber type shift. So your muscles are generally composed of 2 fiber types. You have the type 1 fibers, which we call oxidative. These fibers are rich with mitochondria, for example. And this fiber type is more resistant to fatigue versus the glycolytic fibers, which are powerful but more susceptible to fatigue. So when you unload the soleus because of space flight and that microgravity exposure, you get this slow oxidative to fast fiber type shift. And so what we found is that when the mice came back down from space and we looked at their muscles, they were more susceptible to fatigue. So basically that means that they tire more easily. And we took a look at some muscles from mice that returned to Earth and had, say, acclimated to Earth for another 2 weeks. And we saw a little bit of recovery, but not anywhere close to the normalization. So aside from looking at the biochemical side of things, so we provided the functional data. The next step is to actually link that functional data with some biochemical or some genomic data. So some folks at NASA are actually going to try to correlate some of our muscle functional measures with some changes in gene expression. Aside from that, what we also did is we returned to Florida. And this is really interesting. What they did is they had the female mice come back down to Earth, and then they bred them with male mice. And the offspring, the F1 pups, we actually looked at the muscles from the F1 pups to see if there was any kind of what we call maternal programming. So if the female mouse goes up into space and returns back to Earth, do they pass down any of these effects from spaceflight onto their pups? In this case, we looked at both male and female pups. And what we found is really interesting. It's a little too early to share the details in full with you, but we will follow up on that as well as some other missions with NASA that we've got lined up.